Imagine this. A tiny, tiny mistake. Just one single error in the massive instruction manual for life. And it leads to this devastating weakness, this anemia.
It sounds almost unbelievable, doesn't it? But that's exactly what physician James Herrick stumbled upon back in 1904. He was looking at a patient's blood cells under a microscope and saw these, well, strangely shaped cells. Thin, elongated, almost like sickles.
Right, hence the name sickle-cell anemia. And we now understand the molecular reality behind it. It's caused by just one tiny alteration in the DNA code for hemoglobin, the protein that carries oxygen in our blood.
Exactly. Specifically, in the beta chain of hemoglobin, a single amino acid gets swapped out. At the 6th position, where there should be glutamic acid.
Right, there's vanillin instead. And that one change, that single substitution, it changes everything about how the protein folds and functions.
It really does. It's a stark reminder, isn't it, that the linear sequence of information we inherit directly dictates the complex, three-dimensional functional machines, the proteins that actually run our bodies.
Absolutely. And that's our mission for this deep dive. We want to unpack that incredible, complex machinery. The system that takes the genetic information, the code, and translates it into functional proteins.
We'll be looking at how proteins get their final shape, tracing the key discoveries that link genes to proteins, cracking the genetic code itself, and then really digging into the nuts and bolts of translation.
Okay, so let's start at the end with the final product. Proteins. These things are just fundamental. You know, if you think of a cell as a bustling city, proteins are doing pretty much everything.
They really are. They're the structural components, the enzymes catalyzing reactions, the transporters, the signalers, everything. They make up about, 15% of a cell's wet weight? It's substantial.
And where does all this functional versatility come from?
It comes down to how they're built. Proteins are essentially long chains called polypeptides, and these chains are made up of individual building blocks, the amino acids.
There are 20 different kinds of amino acids, right?
20 standard ones, yes. They link together one after another through what are called peptide bonds. It's a condensation reaction, basically releasing a water molecule each time a bond forms.
And the key is that these 20 amino acids have different properties because of their side groups, their R groups.
Exactly. You've got some that hate water hydrophobic, non-polar ones. Others love water hydrophilic, polar ones. And then you have charged ones, either acidic, which are negatively charged, or basic, positively charged.
So that mix of properties is what allows the protein to do its job.
Absolutely. That chemical diversity dictates how the polypeptide chain folds up into a specific three-dimensional shape, and that shape determines its function. This folding happens in stages or levels of organization.
Yes, the 4 levels.
Right. First, you have the primary structure. That's simply the linear sequence of amino acids in the chain, just the order they're linked up in, dictated directly by the gene.
Okay, simple enough. The basic blueprint.
Then comes the secondary structure. As soon as that chain is made, it starts to fold locally. You get these common shapes like the alpha helix for like a coiled spring, or the beta sheet, which looks like a folded ribbon.
And what holds those shapes together?
Hydrogen bonds. Specifically, hydrogen bonds between the backbone atoms of the polypeptide chain itself, not the side groups. Oh, and an interesting point, the amino acid proline is too rigid. It actually breaks alpha helices.
Huh. Okay, so primary is the sequence, secondary is local folding.
Then you get the tertiary structure. This is the overall, complete 3D shape of a single polypeptide chain. It's how the whole thing balls up.
And this is where those side groups come into play.
Precisely. Interactions between the different R groups dictate the tertiary structure. Hydrophobic groups burying themselves inside, ionic bonds between charged groups, more hydrogen bonds, even weak van der Waals forces. And critically, sometimes you get strong covalent bonds called disulfide bridges between cysteine amino acids, really locking the shape in place.
Okay, the full 3D shape. Is that it?
For many proteins, yes. But some are even more complex. They have quaternary structure. This is when 2 or more separate polypeptide chains, each folded into its tertiary structure, come together to form a functional complex.
Like hemoglobin, which we started with.
Exactly.
Yeah.
Hemoglobin is a great example. It's a tetramer, meaning 4 subunits. 2 alpha-globin chains and 2 beta-globin chains, all working together.
So understanding that incredibly precise final structure really makes you wonder, how did scientists first figure out that genes controlled this exact amino acid sequence?
Yeah, that's a fascinating story. There were early hints, like Archibald Garrod's work connecting genes and metabolism around the turn of the 20th century. But the really definitive link came later in the 1940s.
With Beadle and Tatum. Right, and their experiments with bread mold.
Yes, George Beadle and Edward Tatum using Neurospora crassa. Their work was just brilliant. They basically set out to break the mold's biochemical pathways one by one.
How did they do that?
They used X-rays to induce random mutations in the mold spores. Then, they grew these spores on what they called a complete medium, basically, a rich soup containing every nutrient the mold could possibly need.
So even if a mutant couldn't make something essential, it could still grow because it was provided in the food.
Exactly. But then came the crucial step. They took spores from colonies that grew on the complete medium and tried to grow them on a minimal medium. This medium only had the absolute bare necessities: sugar, salts, one vitamin.
And the wild type, the non-mutated mold, could grow fine on that because it could synthesize all the other amino acids and vitamins it needed from scratch.
Right. But if a mutant couldn't grow on the minimal medium, Beadle and Tatum knew they'd hit a gene involved in synthesizing something essential.
So how did they figure out what was broken?
Systematically, they'd take that mutant that couldn't grow on minimal medium and try growing it on minimal medium plus one specific supplement. Maybe minimal plus amino acid X, or minimal plus vitamin Y. If the mold suddenly grew when they added, say, arginine, they knew they'd mutated a gene required for making arginine.
And because they could often pinpoint a single missing step in a known biochemical pathway,
They concluded that one gene controls one specific enzymatic step. This led to their famous hypothesis: one gene, one enzyme.
a huge conceptual leap. Though we've refined that slightly since then, haven't we?
We have. As we learned more, we found enzymes made of multiple different polypeptide chains, each coded by its own gene, like tryptophan synthetase or hemoglobin itself. So the slogan needed tweaking.
To one gene, one polypeptide, because one gene really specifies the sequence of one single polypeptide chain.
Precisely. It's a more accurate reflection of the underlying genetics.
Okay, so one gene codes for one polypeptide chain. But how? How does the sequence of DNA bases translate into a sequence of amino acids? That's the core puzzle of the genetic code.
It is. You've got only 4 letters in the DNA alphabet: A, T, C, and G, or A, U, C, G in RNA. But you need to specify 20 different amino acids.
So a one-to-one code clearly doesn't work. 4 bases, 20 amino acids. What about pairs of bases, like AA, AU, AC, AG, UA, UU, and so on?
Well, if you calculate the number of possible pairs, 4 times 4 gives you only 16 combinations. Still not enough to code for 20 amino acids.
But triplets, if you use 3 bases per code word or codon?
Then you get 4 times 4 times 4, which equals 64 possible combinations.
Yeah.
That's more than enough to specify all 20 amino acids, plus potentially signals for starting and stopping protein synthesis.
So the hypothesis was the genetic code is read in units of 3 nucleotides called codons.
Yes, and critically, it was thought to be read continuously, without overlaps or gaps. But proving this triplet nature experimentally, that was another piece of scientific elegance.
This involved Francis Crick again, didn't it? And bacteriophages.
It did. Crick and his colleagues used bacteriophage T4, a virus that infects bacteria, and they used a chemical called proflavin. Proflavin is interesting because it tends to cause insertions or deletions of a single base pair in the DNA.
Okay, so tiny little typos, what effect did that have?
Well, think about reading a sentence in groups of 3 letters. The fat cat ate the rat. If you insert just one letter, say an X right after the HE, the X of TCA, tat, eth, eero, T. The reading frame shifts, and everything downstream becomes gibberish.
Right, a frame shift mutation. So a single insertion plus or dilution would likely wreck the gene product, leading to a mutant phage.
Exactly, and that's what they saw. But here was Crick's genius insight. What if the code really is read in 3s? What would happen if you combine 3 single insertions close together?
If you add 3 letters, maybe the XXX sat cat ate the rat. The reading frame downstream is restored.
Precisely. Or if you combine 3 single deletions. And experimentally, that's exactly what happened. While phages with 1 plus or 2 plus plus insertions were mutant, phages engineered to have 3 nearby insertions, plus plus plus, often showed a wild type or near wild type phenotype. The same was true for 3 deletions.
That's incredible. It basically proved the code is triplet and read continuously from a fixed starting point. 3 single shifts cancel each other out, restoring the correct downstream reading frame.
It was definitive evidence. The codon is 3 nucleotides long. Read one after another without overlapping.
Okay, so we've established the code is triplet. Now let's look at the actual machinery that performs this translation. It's incredibly complex.
It really is. You need the mRNA message, of course. But you also need ribosomes, dozens of accessory proteins, several types of ribosomal RNA, plus a whole suite of transfer RNAs and the enzymes that charge them. It's estimated to involve over 50 polypeptides, 3 to 5 RNA molecules, maybe 40, 60 different tRNAs. It's a massive operation.
Before we dive into the process, maybe let's summarize the key features of the code itself. You mentioned there were 7 properties.
Right, let's quickly list them. 1, it's triplet. 3 nucleotides specify 1 amino acid.
Yeah.
2, it's non-overlapping, reads sequentially, codon by codon. 3, it's comma-free. No punctuation between codons within the coding region.
4, it's degenerate. This is important. Most amino acids are specified by more than 1 codon.
Yes, very important. 5, it's ordered. Codons for the same amino acid or similar amino acids often differ by just 1 base, usually the 3rd one. This provides some error tolerance.
6, it has specific start and stop signals. AUG is the usual start codon, coding for methionine, and there are 3 stop codons, UAA, UAG, UGA, that signal termination.
And finally, 7. The code is nearly universal. With very minor exceptions, like in mitochondria, the same codons specify the same amino acids in virtually all organisms, from bacteria to humans.
That universality is pretty profound evidence for a common ancestor for all life.
It really is. Now, the main stage for translation is the ribosome.
The protein synthesis factory.
You could call it that. It's this huge complex made of ribosomal RNA, rRNA, and ribosomal proteins. It has 2 main parts, a large subunit and a small subunit. Prokaryotic ribosomes are called 70S. Eukaryotic ones are 80S, slightly larger.
And critically, the ribosome has specific binding sites for the other key players.
Yes, 3 crucial sites for transfer RNA, or tRNA. There's the A site for aminoacyl-tRNA. This is where the tRNA carrying the next amino acid to be added binds.
Okay, A for incoming amino acid.
Then the P site for peptidyl-tRNA. This site holds the tRNA attached to the growing polypeptide chain.
P for polypeptide.
And finally, the E site for exit. This is where the tRNA, now having delivered its amino acid, sits briefly before leaving the ribosome.
APE. Got it. Now, the tRNA itself, this was Crick's adapter molecule, right? The thing that physically links the mRNA codon to the amino acid.
Exactly. He hypothesized there must be some molecule that could read the codon on one end and carry the corresponding amino acid on the other. That's tRNA. They're relatively small RNA molecules.
And they have that crucial 3-base sequence, the anticodon.
Yes. The anticodon loop on the tRNA base pairs directly in an anti-parallel fashion with the codon on the mRNA. But the tRNA isn't useful until it's carrying its specific amino acid. This process is called charging.
And that charging has to be incredibly accurate.
Oh, absolutely critical. It's carried out by a set of enzymes called aminoacyl-tRNA synthetases. There's essentially 1 synthetase for each amino acid. These enzymes are amazing. They recognize both a specific amino acid and all the tRNAs that should carry it.
Using ATP energy, they attach the correct amino acid to the correct tRNA. If this step goes wrong, the whole system fails.
Precisely. The ribosome relies entirely on the correct pairing between the codon and the anticodon. It has no way to check if the tRNA is carrying the right amino acid. The fidelity rests heavily on those synthetases.
All right, we have the code, the ribosome workbench, and the charged tRNA adapters. Let's walk through the actual process. Initiation, elongation, termination. How does it all begin? Initiation.
Initiation sets the stage. It ensures the ribosome finds the correct starting point on the mRNA and brings in the first amino acid. Interestingly, all polypeptides initially start with the amino acid methionine, though it's often removed later. There's a special initiator tRNA for this.
How does the ribosome know where to start on the long mRNA molecule?
In prokaryotes like bacteria, there's a specific sequence upstream of the AUG start codon called the Shine-Dalgarno sequence. It's typically AGG, something similar.
And this sequence base pairs with
a complementary sequence in the 16S rRNA of the small ribosomal subunit. This interaction perfectly positions the small subunit so the AUG start codon is aligned with what will become the P site. Then, with the help of initiation factors and GTP energy, the initiator tRNA carrying methionine binds to the P site, and the large subunit joins the complex.
So the first tRNA actually starts in the P site, not the A site.
For the initiator tRNA, yes, that's unique to initiation.
Okay. What about eukaryotes? Is it the same?
It's similar in principle, but more complex, involving more initiation factors. Eukaryotes don't use the Shine-Dalgarno sequence. Instead, the small ribosomal subunit, along with the initiator tRNA and other factors, typically binds near the 5-foot cap of the mRNA.
The cap structure at the beginning of the eukaryotic message.
Right. And then the complex scans along the mRNA, moving downstream until it finds the first AUG codon in an appropriate context.
Ah, that context is important, isn't it? Kozak's rules.
Yes, Merrill and Kozak found that the sequence surrounding the AUG influences how efficiently it's recognized as a start codon. The optimal sequence is something like 5-GCCA or GCCOG3. Once the right AUG is found, the large subunit joins, again using GTP.
Okay, initiation complete. Ribosome assembled the start codon, initiator tRNA in the P site. Now for elongation, building the chain.
Elongation is a cyclical process with three main steps, repeated over and over. And it involves soluble protein factors called elongation factors, plus more GTP energy.
Step 1.
Step 1. A charged tRNA carrying the next amino acid specified by the codon in the A site enters the A site. This requires an elongation factor called EF2 in prokaryotes, bound to GTP.
So the correctly charged tRNA slots into the A site, matching the codon there.
Right. Step 2, this is the crucial chemical reaction peptide bond formation. The amino acid or peptide chain attached to the tRNA in the P site is transferred and covalently linked to the amino acid on the tRNA in the A site.
Forming a new, longer peptide bond. And this is catalyzed by...
It's catalyzed by peptidyltransferase activity. And here's a mind-blowing discovery. This catalytic activity is not performed by a ribosomal protein, but by the 23S ribosomal RNA in the large subunit.
Wow. So the ribosome is actually a ribozyme, an RNA enzyme at its core.
Absolutely. The fundamental reaction of life, building proteins, is catalyzed by RNA. It's a huge piece of evidence for the RNA world hypothesis about the origin of life.
That's amazing. Okay, so the bond forms, the chain is transferred to the A site tRNA. What's step 3?
Step 3 is translocation. The ribosome literally moves 3 nucleotides down the mRNA towards the 3' kinase. This requires another elongation factor, EFG, and GTP hydrolysis.
And what does this shift do to the tRNAs?
The tRNA that was in the A site, now carrying the elongated polypeptide chain, moves into the P site. The tRNA that was in the P site, now when charged, moves into the E site.
And from the E site, it exits the ribosome.
Exactly. And now the A site is empty again, exposing the next codon, ready for the cycle to repeat. Bind a new tRNA, form the peptide bond, translocate over and over.
I know.
Until one of the 3 stop codons, UAA, UAG, or UGA, enters the A site. This signals termination.
And there are no tRNAs that recognize these stop codons.
Correct. Instead, proteins called release factors, RFs, recognize the stop codons when they arrive in the A site.
What do the release factors do?
Binding of a release factor essentially modifies the peptidyltransferase center. Instead of catalyzing the addition of another amino acid, it promotes the addition of a water molecule to the end of the polypeptide chain.
Which breaks the bond connecting the polypeptide to the P site tRNA.
Precisely. It hydrolyzes that bond, releasing the completed polypeptide chain from the ribosome. Then other factors help the ribosomal subunits, the mRNA, and the release factors to all dissociate, ready to start translation again on another message.
So the process is incredibly specific, relying on codon-anticodon pairing. But you mentioned earlier the code is degenerate, and that seems related to flexibility. Tell us about the Wobble hypothesis.
Right. Crick proposed the Wobble hypothesis after the genetic code was largely deciphered to explain some patterns they saw. The degeneracy means, for instance, that an amino acid like alanine is coded by GCU, GCC, GCA, and GCG. 4 codons, 1 amino acid.
Does the cell need 4 different tRNAs for alanine?
Not necessarily. That's where Wobble comes in. Crick suggested that the base pairing rules between the codon on mRNA and the anticodon on tRNA might be strict for the first 2 positions of the codon, but less stringent, or wobbly, for the 3rd position.
The 3rd base of the codon pairs with the 1st base of the anticodon, right? Because they run antiparallel.
Exactly. So the wobble occurs at the 5-foot base of the anticodon. This allows a single tRNA anticodon to recognize more than one codon.
Is there a molecular basis for this?
Yes. For example, sometimes the base at the wobble position, 5-foot end of the anticodon, is a modified base called inosine. Inosine is structurally similar to guanine, but lacks an amino group. Because of its structure, inosine in the anticodon can actually form hydrogen bonds with U, C, or A in the 3rd position of the codon.
Wow. So one tRNA with inosine at the wobble position could recognize 3 different codons ending in U, C, or A, all coding for the same amino acid.
Precisely. It explains how fewer tRNAs are needed than the 61 sense codons might suggest. And it adds another layer of robustness to the codon. Changes in that 3rd codon position often don't change the amino acid.
That makes sense. Now, what about when things do go wrong? You mentioned suppressor mutations. How can one mutation suppress another?
This is another fascinating aspect of genetics. Let's say you have an initial mutation, perhaps a nonsense mutation. That's one that changes a normal codon into a stop codon, UAA, UAG, or UGA.
Like the UAG codon, often called an amber mutation. That would normally cause premature termination, right? A short, probably non-functional protein.
Correct. But sometimes a second mutation can occur elsewhere in the genome that partially or fully restores the normal phenotype, even though the original nonsense mutation is still there. This second mutation is a suppressor.
How does it work?
One common type is a suppressor tRNA. This is a mutation in a gene that codes for a tRNA molecule. The mutation changes the anticodon of the tRNA
So that it now recognizes.
so that it now recognizes the stop codon. For example, a mutation might change the anticodon of a tRNA that normally carries tyrosine so that it now recognizes the UAG stop codon.
Ah, so when the ribosome hits that premature UAG stop codon, instead of terminating, this mutant suppressor tRNA binds and inserts tyrosine, allowing translation to continue.
Exactly. It suppresses the effect of the nonsense mutation by reading through the stop signal. Now, the resulting protein might have a wrong amino acid, tyrosine, instead of whatever was originally there, but it's often full length and might retain some function, which is better than a truncated protein.
So the genetic system even has ways to sometimes bypass its own stop signs if needed. Remarkable flexibility.
It really shows the interplay and sometimes the error correction or error tolerance mechanisms built into gene expression.
This has been a really comprehensive journey. We started with sickle cell anemia, that single amino acid change with devastating consequences, which really highlights the importance of protein structure.
Yeah, and we saw how that structure builds up from the primary sequence through secondary folds like helices and sheets to the overall tertiary shape dictated by side chain interactions and sometimes quaternary assembly of multiple chains.
Then we traced the history, the crucial link made by Beadle and Tatum with their one gene one enzyme idea, later refined to one gene one polypeptide.
Followed by Crick and colleagues elegantly proving the triplet nature of the genetic code using those clever frameshift experiments in phage T4.
We laid out the 7 key properties of that code triplet: non-overlapping, degenerate, ordered, start-stop signals, and nearly universal.
And dissected the machinery. The ribosome, with its crucial A, P, and E sites acting as the workbench, and the tRNA molecules as the adapters, charged accurately by the aminoacyl-tRNA synthetases.
We walk step by step through initiation, finding the start signal using Shine-Dalgarno or scanning mechanisms, elongation, the cycle of tRNA binding, peptide bond formation catalyzed by RNA, and translocation, and finally termination, triggered by stop codons and release factors.
And we touched on the nuances of the Wobble hypothesis explaining degeneracy in tRNA economy, and suppressor tRNAs showing how the system can sometimes even overcome errors like premature stop codons.
The whole process hinges on incredible specificity, especially in codon-anticodon pairing and tRNA charging. Yet, built into this precision is this amazing flexibility, this degeneracy provided by Wobble, which acts almost like a buffer against mutation.
It really is a system optimized for both accuracy and resilience.
Yeah.
It translates the genetic blueprint into the working parts of the cell with remarkable fidelity, but it also has ways to cope with the inevitable errors that creep in.
Which leaves us with a final thought for you, our listener. We emphasized how crucial the aminoacyl-tRNA synthetases are. They are the gatekeepers, ensuring the right amino acid is attached to the right tRNA family. The ribosome itself just trusts the tRNA.
So consider this. Given the catastrophic consequences, if a synthetase consistently made mistakes putting alanine on every tRNA meant for glycine, for example, what kind of proofreading or quality control mechanisms do you think must exist within those synthetase enzymes themselves to achieve such extraordinary accuracy, often better than one mistake in 10,000 charging events? What internal checks might be happening before that charged tRNA even reaches the ribosome?
Something to ponder about the layers of fidelity required to build life correctly. Thank you for joining us on this deep dive into the heart of gene expression.